Nee. Oh.
Jij is van de rente.
Thanks, everybody, for coming back. Welcome. The second part of the lecture on. Is the screen readable, or should I turn off the lights on the screen? Yes? Should we turn off? Switch for that. Yeah, that's slightly better. Okay. Now it's warm. Was it not too cold outside? No. You guys were in the sun most of the time. Yeah. Okay. Yeah. So this is where we left off, right? So now we can actually we can count the number of minor sits in a column, calculate the frequencies, and then we can calculate the information entropy in the column, and that gives us a measure of the conservation in that column. But now you understand why those two things are related. Very good. So just to give you a bit of a feeling of how this score works. We have here a set of different alignment columns with two amino acids types, alanines and leucines. So there's two probabilities involved there. There's the probability of observing an alanine in a column and the probability of leucine. An alanine goes from 1 over here to 0 over there, and leucine the other way. So not very fancy. And then you calculate the information entropy by doing P log P. And then you get this curve, as I already mentioned, when the entropy is 0 when there's no choice, no information gained from an additional experiment, which means over here when there's only alanines, so a new sequence will be an alanine. We don't learn anything new. The same on the other side, and the amount of uncertainty or the amount of information gained from more experiments is maximum when the probabilities are equal in the middle. where there's 50% alanine and 50% leucine. Doesn't matter how many different types you have. Now there's 2, but if there's 3, then you have the maximum where they're all 1 per except then you can't really easily do this in a 2-dimensional plot because you have 3 frequencies, so it gets 3. Sorry, this is 2 frequencies. It's 1-dimensional because I have to add up to 1. But if you have 3 frequencies, then there's 2 dimensions, right? So you can't easily afford it. You have a 2-dimensional. Okay, so this is what I just said. Now you can go back to the example that's in the slides, which I'll skip over for the sake of time. So, but then conservation is the easy case, right, in terms of biology, because and it's sort of the obvious thing, right? If there's selective pressure, then you'll on a particular amino acid in a particular position, then you'll observe that this position becomes or has become conserved. But there's much more to it than we have. There's much more that we usually have if we're looking at a protein family. We're not looking at individual proteins at this point, right? We're looking at protein families. But if a typical large protein family has subgroups which have slight variations on the function. Maybe they have a different substrate specificity, so they bind different substrates, or they have a different preference for different substrates, or maybe they're regulated in a different way. Lots of other. So if we go one step beyond conservation, where we just look for the, I would almost say, blindingly obvious things that are important, then what's the next step? If you have not just one, let's say, monolithic protein family where you can assume that everything, all the function is completely conserved throughout the whole family, it's actually quite unlikely. If you have certainly if you're looking at the use diversity in evolutionary history. Right? So what's the next step? Where would you look? If you know that there's some variation in some diversity within subgroups in your protein family, that might tell you more about the function. Yeah. You might want to compare these subgroups, right, and then see. But what are you looking for then? How do you find the residues? What would you look for to find residues that are important for this variation between subgroups? Your brains are still warming up from sitting outside. So regions that have high variability, but then that sounds like it's unconserved. So there's sort of we have 3 situations now, right? We have a conserved position, which is going to be, if it's conserved throughout your whole protein family, it's not going to be different between subgroups. You have an unconserved position, which might be different between the subgroups, but it's different anyway. So that's also not so very interesting in terms of explaining the functional differences between these subgroups. So what are you looking for?
Positions that are conserved in one and
They're conserved in one and
not in the other.
they're not in the other. So, like, let me write an example. So you have, now I have 3 sequences, but there's a subdivision, right? And let's say we have a tryptophan in one position in the one subgroup, and then it's different in the other. It's not conserved. That's what you meant? Yeah. Sorry, I don't remember exactly who said this, but yeah. You also like you're thinking of something similar? Yeah. Okay. Now, so we have these 2 subgroups. So now you can imagine that this conserved tryptophan is very important for the function of the top. But what about the bottom group?
No, it's not. They're all small hydrophobic residues. So apparently you only need small hydrophobic residues.
Okay, so there's some conservation here if you know your amino acids, right? These are small aromatic, not aromatic, small hydrophobics. But what if it is
now it's random, right?
so is this going to be important? Well, for group, let's call this group A. For group A, yes, maybe. So what would be the, if we're talking about completely obvious things, what would be the obvious one that's very important for the difference between these two groups? Let's say we have really doesn't matter which one I choose. If you have something like this, right? That's going to be the obvious case, where you have conservation on both sides, but they're different. And you can do the same kind of logic here as you do for the conservation, where you count the number of ways in which you can get a different arrangement of amino acids over two groups of sequences in this position. That's not how I build it up, because that takes way too much time. Oh yes, I did this. And so, but we're looking now at differences in these subgroups. And so what actually, there's a couple of methods that do that. There's, I don't remember, I think there might be a slide somewhere later on for reference that lists some of those methods. And then you look for, the methods to look for conserved sites within a subfamily, but they're very strict. They aim to be conserved. They won't even consider this case as being somewhat conserved, because they'll just say, oh, they're different. Actually, it turns out that this case is much more common than this one. Because what you often have is things about specificity, where there's this residue might be, so if specificity is usually about binding, right? So if this tryptophan is very important for binding a particular ligand for the subfamily A, then, and maybe it's important for subfamily B to not bind the same ligand, maybe because they're in the signaling pathway and they shouldn't listen to each other's signals. Then you can make the specificity by doing something very different here, but it's easier, in an evolutionary way or sense of looking at things, to have a different position where you say, okay, well, if I have a tryptophan here, I can bind this ligand. If I not have a tryptophan here, I can't bind it. Then I'll put, but if I put a serine here, and this is different position in the sequence, but maybe it's the same binding area on the surface of the protein, in the three-dimensional structure of the protein. I need a serine there to bind the ligand for B, and just anything else, just not a serine, or maybe not a polar amino acid. So maybe I need something hydrophobic there to avoid binding ligand B. Yeah? So this one will bind A, and this one bind B, and this one won't bind A, and where small a and b are the ligands, right? That's the idea. This is what you see a lot, actually. Okay, so how do we do that? Oh yeah, just to put some numbers to my statement. There's a couple of protein families that we looked into when we were starting to look into this specificity, and we actually, for these 3-4 cases, there are actually experimentally verified specificity positions. So positions where it's known that there is, first of all, it's known that there are functional differences within these protein families that are important for function. This one is a component of a signaling cascade where a receptor gets a signal from outside the cell. The SMOT is the cytosolic protein that transfers the signal to the nucleus, and then there are homologues of these whole pathways. So there's homologues of the receptor, and there's homologues of the transcription factor in the nucleus. And these SMOT proteins have to recognize the right receptor and not the other one, and have to recognize the right transcription factor in the nucleus and not the other one. And so they know which positions are important for this specificity. Yeah.
I was wondering, if you take one column, just one column in regard, that's not very strong. Yeah, in this case,
Yeah.
but how many columns do you take for such a experiment? How many different?
Well, we look at the whole alignment, but we look at one column at a time. I'm not sure that answers your question.
Yeah, but I mean, can you see from that point? I mean, if you look at several columns.
Why not look at multiple columns?
Yeah, I mean.
Well, if you only have the sequence, this is difficult because which columns to combine.
Exactly.
If you have 100 residues in your alignment, your 100 positions, then you have 50,000 possible pairs of columns that you might want to group together, right? So so without further information about which ones to take together, if you have a structure, it's easier because then you can just take the neighbors in the structure, so the columns in your sequence that corresponds to neighbors in the structure. Some of them will be neighbors in the sequence, but others will be from different parts of the sequence. Then you can but if you don't have the structure information, that's very difficult.
Okay, so you need some additional.
Okay, so anyway, for all of these protein families, there's known specificity which has a functional importance, like I just sketched for this SMOT protein in the signaling cascade. And then you look in the alignment where how the conservation is between the subgroups on these known positions. So these are not just positions that are known to be important for the function. These are positions that are known to be important for the differences in function between the subfamilies. Right? And then you see, so there's three possible cases. Either they're conserved in all the subgroups, or they're conserved in only one, or they're not conserved in either subgroup. And then you see that out of about 100 known sites in total, only about a quarter are conserved on both sides. About half are only conserved on one side, and there's this situation where it's conserved on one side and variable in the other, but different. And there's also some cases where they're not conserved on either side, but they can still be different, because you can still have something like you can have small hydrophobic on one side and aromatic on the other side. Yes. So the reason why we looked into this is because the 5 or so existing methods that we looked at, they all require quite strict conservation on both sides, and that means that that you're only finding this quarter of the sites that you're interested in. You're not finding either, and you're certainly not going to find those, and they're still important. And so while conservation is very important, yes, conservation is important, because if a position is conserved, you know it was important for something, because otherwise it would have changed. Yes, that's where positive selection comes from. There's two problems with conservation. First of all, you don't know what it was conserved for. Presumably the function, but the function has different aspects. It could be the stability of the protein. It could even be something about the messenger RNA secondary structure, that you don't want secondary structure in messenger RNA. You change it. Maybe you get secondary structure and that breaks the ribosome going through the translation. Or it could be It could be some part of the function that you're not really interested in. If you're interested in ligand binding, but this position is important for some regulatory regulation of your enzyme, then it's telling you a different story. But then, so that's one part of the problem. You know it's important, but you don't know exactly what it's important for. The other part is that it doesn't mean that all the other positions that are not conserved or less conserved, that they're not important. It just means that they haven't been strongly positively selected for. But they're still going to be there, and they're going to do something. Yeah? Now, in this case, but it's used a lot because it always works if you don't know anything else about your protein. You can look at the conserved sites and say, well, these are going to be important, and you're going to be right. But if you know that you have subfamilies, then you can look at the differences, and then suddenly the conservation becomes less important. If you see differences, that's your signal. If you see conserved differences, it's a stronger signal, but differences are still going to be important for the difference in function for your two protein subgroups in your protein family. Okay. So now we need a measure to do that, and I won't go through the derivation, but we've done the sequence entropy thing, and if you're talking about the way we derive it from the faculties, that's actually just combinatorics. And from combinatorics, it's a small step to conditional probabilities or Bayes statistics, where you say, what is the probability of observing this when you already know that? Yeah. So we can apply this to our wrong button. We can apply this to our alliance. So what we did before was we have 5 sequences where there's all alanines, and we say, okay, we already know there's alanines here. We take our 6th sequence, and we're not surprised if there's again an alanine, because that's what we already saw. Now you can do this between groups as well, and then you're going to look at the amino acids that you observe in group A, and then you're going to look at group B, and then you say, am I surprised by what I see in group B? Does this give me any new information? If it does, then you've learned a lot. There's a high information content in group B. If not, if you have a position that is something like this, yeah, you've observed group A, then you're going to look at group B, like, oh, well, nothing new there, right? So this one isn't going to be very interesting for explaining differences in group B. The conserved side is definitely not going to be interesting. Yeah, we get that. This one might actually be interesting. This one is certainly also going to be interesting. Yeah? Okay, so now we can look at conditional entropy or conditional probabilities to have a measure for that. And it relates to Bayes statistics, and I'm not going to go into all the details there. But the formula it's similar to the one that we already had, but now we're looking at the entropy of the combined event, and we have probabilities of combined events. So the probability of observing J when I already happened. So the probability of observing an A in group B when you already know the frequencies of the amino acids in group A. And you can work this out. I'm not going to go through the details here. Oh yes, I should do actually. Oh yes. So the thing is that you can rework this. Oh right, yes. So now what we do is this is the information. How to say that? It's the conditional entropy of observing group B when you know group A. Let's just look at this column, not too confusing. But you can also calculate the entropy in each of the groups. So, I have to say it right. So, sorry. This is the entropy of the total column, the information content of the total column, because it's the combined events in group X and Y for events I and J, where I and J are the different amino acids that you observe. But you can do it for each group separately, and for the other one group as well. And now, what you're interested in is whether the total information in the whole column is more than the sum of the two groups. Yeah? Because if it's the same, then there was no information gained from combining the two groups. There's nothing new in the other group. If the information in the total column is higher, because there's more different stuff there to observe, that means that you can't learn the whole column by just looking at A or B. There's something new if you look at the other group. Then you know that you have something. And so the total information content can only be smaller than the sum of the two, right? So if you, am I getting you guys confused now?
Yeah.
Yeah. That's because I'm confused. How does this work?
An example would be better.
Hmm?
An example, give me an example.
Yeah, I'm trying to work out the example in my head, but it's I've lost the bed somewhere. Some. It doesn't add up in my head. Because I'm stuck on the wrong track. Because if I take the entropy of this, it's very high, and the entropy of this is very high as well. Oh right, now I get it.
There's a minus in H, so if you add two things and they should be more than the sum, then the H of the combined one will be smaller than the Hx and Hy. Right? Written wrong here.
Yeah.
Combined entropy is larger than minus.
Yeah,
So it's smaller.
so it's not.
Combining entropy makes it more negative.
Yeah.
With more.
Okay, but I got it here. So the entropy here is high, the entropy there is high, but the combined entropy is actually the same. Because it's the maximum variation that you get. You have three sequences, they're all three different. Three sequences are all three different. But now you have six sequences and they're all six different. Yeah? So that works out to be the same entropy. Whereas here, this is fairly, I can make it even nicer. Let's do this. Now we have three sequences, they're all three different. Maximum entropy. Three sequences, they're all three different. Maximum entropy. If you combine them, there's six sequences with three different amino acids. So the entropy is a lot lower. Yeah? So in this case, you can say, well, they're not independent. They have the same. So there is a bit of conservation here, which you actually only gets clear when you look at both groups. Oh, they're all polar. So that means if you combine these two groups, the total information is relatively less than when you look at the independent groups. Yeah? That's what you get here. Then they're dependent. So it's like throwing a die. So let's say you have a six-sided die. you throw it 3 times, you get 1, 2, and 3. That's normal. If you throw it again, you get 1, 2, and 3. Maybe that it could still be normal, right? It's only 6 samples. It's too little. But if you throw it 100 times, you get 1, 2, 3, and another 100 times you get 1, 2, 3, and you know there's something wrong. Yeah, there's a correlation there. Yeah. But if you get different things, or if you throw it 3 times, 1, 2, 3, and then another 3 times you get 4, 5, 6, yeah, then there's no correlation. So this is a way to measure the dependence or independence of these 2 events. And if they're dependent, then then it tells you something about conservation. So they're still important, these positions, but not for the differences. Yeah? If they're independent, then they've evolved under different selection. Yeah, because that makes them independent. Yeah.
But if one position is conserved in A and then with another amino acid conserved in B,
Yeah.
you would say that the entropy in A and B is very low, but then together it would be higher. So then that wouldn't make sense.
So it's a negative in the formula. So
But then the first explanation is
Yeah, but the logs are negative because it's fractions.
yeah.
So this is always a positive number. Yeah. So if you have this, so what you're saying, right? So the entropy is 0, or 0 within the group, but for the whole group, it's actually it's not 0. So
those are the positions you're interested in, right? More than
it's definitely one that you're interested in. So this is the source of my original confusion, and so I haven't quite solved it. I don't know. I'll have to work it out. I'll come back to it, but not today. At least not before the end of the lecture. Okay. I need to see where we're going. Oh, yes. Right, yes. Okay. So this is the formula from, I'm sorry, then I can't answer your question right now. But I'll come back to it. But you might have to remind me. So this is the formula for conditional entropy, where you're looking at the event A, an event in group A, when you already know the event in group B. So this is how it works out. Oh, sorry, it's the other way around. You look at the frequencies in B, given that you already know those in A. So you say, oh, I see an histidine here, and how remarkable is that, given the fact that I also saw a histidine here. Or I see a valine here, given that I haven't actually seen a valine in group A. So now if you have a few examples, so it looks similar to the other one that I had, but now we have 2 groups with, to keep it simple, alanines and leucines, where we have different fractions of alanines and leucines in group A, and then no or a few leucines in group B. We can do this 2 ways. That's the silly thing about this conditional entropy, because we can do B versus A or A versus B. And so if you, well, this one is simple, it's 0. Let's look at your, yes, sorry, got sidetracked. No, this is your case, right? Where you have some conserved here, and so the problem with this conditional entropy is that whenever you have a frequency of 0 in the other group, if your probability of a leucine in group B is 0, it goes, it's dividing by 0, it goes infinite.
Yeah, I'm a little bit confused because at part A you have the first one that's only A's, so that's 0 entropy.
Yeah.
And if you go to the right, at one point you have only L, and then it's not 0, but you don't have any choice, so I don't get why that's not entropy 0.
Because we're looking at the conditional entropy here, so it's about the information gained when including information from the other group.
You gain a lot of information.
Because you're seeing things that you haven't seen before. So the information gain here is 0 because it's more alanines. Yeah? Whereas the information gain here, it's a bit higher because you've seen some leucines. So you expect, it's a small information gain because you expect an alanine, but every time you see it, you know that it's not going to be a leucine. It could have been a leucine, right? And then over here, the formula actually breaks down because the information gain is infinite, which is not very convenient one. So this formula, it sort of works, but it gets infinite for the positions that you're actually interested in. Also interesting to point out, so over here we actually have a mix of alanine and leucines, and because it's 2/3 alanine and 1/3 leucine, and because we see exactly the same proportions in group B, there's exactly no information gain. You see an alanine and a leucine, you look, oh yeah, well, that's what I already have. Yeah? But it's 0 here, so that covers that quite nicely. But it's inconvenient if it goes, and it's also very asymmetric, because if you compare A with B, it already goes infinite here because you say what? So A with B, then no, the other way around. If you this is A with B. If you compare B with A, then you don't have any frequency for leucine in group B. So you divide by 0. So this makes it very inconvenient to use in practice. There's tricks around that, but I don't want to go into that here.
For leucine or for alanine?
Sorry?
Are these for leucine or for alanine? These graphs?
No, this is the sum. So you sum over X, which is all your amino acids. And then for all the other ones that you don't have, the 18 others, you also have zeros here, but then there's 0 on both sides. And you can derive that the logarithm of 0 divided by 0 is actually 0. It doesn't have to be because you're dividing by 0, but since you're 0 divided by 0, it can be, and it turns out that it is. But that's too far. I don't do that derivation. Yeah.
I have a question about the case where you have in B the LAA and in A you have the LLAAA. You say then that
Which one?
Yeah.
This one?
Then you compare two probabilities, but I do not see how you make one a condition for the other probability.
I haven't shown you why that is the case, but using this formula, that's what you do. So this formula is the conditional entropy of the probabilities in group A when you already know them in group B. But the other way around, I will get them mixed up. So it's on the previous slide. Yeah. Okay. You can actually normalize this in one way to get rid of the one easy trick to get rid of the infinites is to not take the probability in group B in the denominator, but the total probability, because if you have an amino acid in group B, let's say leucine, then you don't have it in A, but you have it in the whole column, right? So then this one is only 0 if this one is also 0. So that solves your infinity. it doesn't solve your asymmetry. We actually did a different trick where we took the sum of those two probabilities. Don't ask me why, but that's what we did. And then, because of the way you do logarithm, it actually flips around. And now you get the nice thing that it actually is between 0 and 1, and it's 0 for the positions where you're interested in. It's still slightly asymmetric, and that you can solve by taking the average of this. So if you do that, so you have, okay, so this was we call this sequence harmony, and this is the one where you do A versus AB, and this is the one where you do B versus AB. And you finally calculate the average of those. You can actually work out that, because this is the definition of the sequence harmony that's on the previous slide, PA log PA over PA plus PB, and the other way around, B, B, A, B. You can split this out if you know your logarithms. You have PA log PA plus PB log PB minus PA log PA plus PB minus PB log PA plus PB, and then you get this, right? But you have PA log PA plus PB minus PA, so you can actually take this PA and that PA together. So now you have PA log PA plus PB log PB minus PA plus PB log PA plus PB. So this is your group A entropy. This is your group B entropy, and this is not quite, but something very close to your total entropy in the whole core. So remember the slide where the confusion was, where the total entropy can be equal or smaller than the sum of the entropes of the groups. If you subtract them, then your value that you have is actually the amount of information difference between the whole core and the sum of the whole core and the sum of the individual groups. So that's how this works. Okay. So this is how it looks, and I have some use cases where we look at how this works in practice, but that is too much to go through now. I do want to show you the rock plots. So this is the sequence harmony. We also did a different method in the same paper, which is called multi-relief. And these are the other methods that we compared with: Proust, XDAT, STP-PRED, and protein keys. Right now there's a couple of newer ones. This is a paper from 2010. So there's new stuff coming out that we haven't really followed up on this. And you see that, well, the differences aren't huge. Protein keys is clearly below the others, but the rest is sort of quite similar. You might say that XDAT is on top here, but if you do the precision recall plot, you can see some bigger differences at the low recall, where sequence harmony and XDAT actually they reach a very high precision at the low recall. So, and then it depends on what you want your predictions for. If you have, let's say you're looking at a protein family, and the validation experiments are very expensive, then you like to have a few predictions that are very accurate, maybe 3 positions where on average you expect 75% accuracy, so 2 out of those 3 will be correct. So you do 3 validations and you get, if you're lucky, 2, maybe 3, but quite likely at least 1 of them will be correct. In other cases where your next step might be a fairly cheap screen where you can easily run hundreds of predictions or validations, then you'd like to have a method, but you can't do everything. Then you'd like to have a method that gets a high coverage, and it's okay to have a low accuracy. You just want to sort of get get rid of maybe 2/3 or 3/4 of your data, and you can focus on, you just don't want to avoid making false negatives. You don't want to throw things away that you should keep, but then you can filter it in the second step. So it depends what your next step is. But sometimes having this high accuracy, even at very low coverage, is a very good thing. And then, so XDAT starts up, so this is us, the blue line. XDAT starts up with us, but it drops down to low precision quite rapidly, and we stay out a little bit longer. Yeah? So one of the reasons that we didn't, or actually the main reason that we didn't continue with this work, is that by this time we've exhausted our nicely curated validation data. That means if you start the continued development of your method, you're starting to train on your validation data. If you don't have any new data, you really cannot test it independently. So that's still the status since 2010 that it's very hard to get reliable data to be able to do a good validation of this method. I've spoken to most of the authors of the other papers over the years in conferences, and they're all in the same position. If there's no more data, you can't make better. Okay, are there any questions about this? No, there's more reading material in the papers and on the slides, but this is the main part of it. Thanks, and see you in a few minutes.